Dear students, today we are going to discuss about the 11th botany, chapter 7, cell cycle. First question, differentiate cytokinesis in plant cell and animal cell. Cytokinesis in animal cell, it is a contractile process. A ring consists of a bundle of microfilaments assembled from actin and myosin. This fibril generates a contractile force. This draws that ring inward, forming a cleavage furrow in the cell. Thus it divides the cell into two. But in the case of plant cell, division of the cytoplasm often starts during the dealer cell phase itself. In plants, cell plate grows from center towards the lateral walls. Likewise, plasmablast contains microtubule, actin filament and vesicle from Golgi apparatus and the endoplasmic reticulum. Microtubules of the plasmablast moves to the equator, fuse to form a new plasma membrane and material which are placed there become new cell wall. The first stage of cell wall construction in a line dividing the newly forming cells called a cell plate. The cell plate evenly stretched right across the cell, forming the middle lamella. Cellulose built up on each side of the middle lamella to form the cell wall of two new plant cells. These are the differences. Define cell cycle. Mention the duration of different phases in human cell cycle. Cell cycle means a series of events leading to the formation of new cell is known as cell cycle. A series of events includes several phases. In human beings, you see that G1 phase, S phase, G2 phase, M phase. The first phase, G1 phase, takes 11 hours. Like that, S phase synthesizing phase 8 hours. Likewise, G2 phase 4 hours and mitotic phase 1 hour. This in human beings cells. Differentiate closed and open mitosis. In the case of closed mitosis, the nuclear envelope remains intact and the chromosome migrate to the opposite pole of a spindle within the nucleus. For example, many single-celled eukaryotes including yeast and slime molds. Likewise, in open mitosis, the nuclear envelope breaks down and then reform around the two sets of separate chromosomes. For best example for this, most of the plants and animals. What is known as crossing over? Here, in the meiosis 1, prophase 1, packaging stage, synapsis is completed and recombination nodules appear at a site where crossing over takes place between non-sister chromatids of homologous chromosomes. Recombinations of homologous chromosome is completed by the end of the stage, but chromosomes are linked at the site of crossing over. This is mediated by enzyme recombination. So, exchange of chromatid between the non-sister homologous chromosomes. Write the significance of meiosis. The first one, this maintaining a definite constant number of chromosomes in organisms. Likewise, crossing over takes place and the exchange of genetic materials leads to variations among species. These variations are the raw material to evolution. Meiosis leads to genetic variability by partitioning different combinations of genes into gametes through independent assortment. Like third one is adaptations of organisms to various environmental stress. Differentiate between mitosis and meiosis. Under mitosis, this here one division takes place, here two divisions. Under the mitosis, number of chromosomes remain the same. Here, number of chromosomes is halved. Likewise, homologous chromosomes line up separately on the metaphase plate. Like that, homologous chromosomes lines up in pairs at the metaphase plate. Here, homologous chromosomes do not pair up. Here, homologous chromosomes pair up to form bivalent. Here, chiasmata do not form and crossing over never occurs. Here, chiasmata form and crossing over occurs. Here, daughter cells are genetically identical. Here, daughter cells are genetically different from parent cells. Here, 2 daughter cells are formed. Here, 4 daughter cells are formed. Give an account of G0 phase or G0 phase. Some cells exit G1 and enter a quiescent stage called G0 phase, where the cell remain metabolically active without proliferation. Cells can exist for a long period in G0 phase. In G0 phase, cells ceases growth with reduced rate of RNA and protein synthesis. The G0 phase is not permanent. Mature neurons, skeletal muscle cells remain permanently in G0 or G0 phase. Many cells in animal remain in G0 phase unless called on to proliferate by appropriate growth factors or other extracellular signals. G0 cells are not dormant.